So say you found a skeleton. How would you tell if it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
All right. Well, hello everyone, and welcome back. So we are not doing our normal Thursday stream where we watch a show and comment on that. Instead, we have a guest today. But first, we have my host and co-host Peter, who is producing the show for us as always.
Back screen.
And we have, he's in the back screen in the background behind the scenes. We also have the one and only Dapper Dinosaur.
Hello.
The Sauropsid, the myth, the legend.
That is all true.
And then we have our guest, whose name is Terry. Welcome.
Hello, hello.
Hello.
Welcome to the show.
Thank you for having me.
So let's see. Okay. All right. Why do I have ads? Why are there so many ads? There are far too many ads.
Because people want your money.
True. I can't have it because I don't even have my money. All right. So as I said via Twitter, we'll give you the floor first, Terry. You can tell us tell us a little bit about yourself, and then the argument you would like to lead with, if you would.
All right. Well, of course, I go by Terry, and I'm a college student right now. I'm studying physics as my major, and I'm just really interested in a lot of different sciences and such. And I am very, very interested in a lot of the new science that's been coming out recently that is very counter to a lot of the things that we were taught in like high school and middle school. And I just kind of wanted to talk about that with you today, and maybe you wanted to talk with me about it today. I don't really know which is which, but I just have a few topics written down that we can start with if you really want to, and we can go from there if you'd like.
Yeah, sure. Whichever one you'd like to go with. We give our guest the floor first. So.
All right. Well, the first point that I wanted to go over, or at least get you guys' opinions and facts on, was like the complexity of life arguments, and a lot of the science and math that's been coming out involved in how complex life really is, all the way down to the molecular level, and a lot of it seems to be very counter to kind of the paradigm that we have always been taught in schools of like chemical evolution and like abiogenesis, for instance. And I seem to think that it lacks a whole lot of scientific backing now. I don't see a whole lot of scientific evidence for those things anymore. But yes, so that's kind of my position on that one.
Well, neither of us, or neither Dapper nor I, are chemists or biochem. We don't typically do origin of life stuff, so I'm not really familiar with the literature in that area, I'm afraid. Sorry.
Well, I do have a question, though, because I do hear this idea of an argument from complexity as some kind of like problem for the theory of evolution, or various attempts at a hypotheses around abiogenesis, and I don't really understand it. I don't know, like, is there a cutoff? Like, this much complexity is too much, and if there is, how do we know that that's where the cutoff should be? Is it a continuum problem? And to me, it just seems like because I never really get those things answered. To me, it sounds like people saying, "Well, that car is too yellow to have won the race." Well, I don't see how that connects, and so I don't see how a complexity connects directly to a problem for evolution or abiogenesis or anything.
Well, I can explain that a little bit, and so you are kind of right that it does have to do with a sort of quote unquote cutoff point, because it more or less has to do with functionality. And so the common argument that you have probably heard is like the mousetrap argument, is that we have mousetraps, and mousetraps have a certain amount of parts, and each part has a specific function. And without all of those parts together simultaneously fulfilling their function, it's not a mousetrap. And it's essentially kind of the same argument with a lot of biological systems, because nowadays we're finding a lot of these biological really machines that are for lack of a better word, what many biologists and even creationists will say is that it's irreducibly complex, meaning that you can't reduce it any further without it fulfilling its function. And there are many examples of that, especially on the bacterial / cellular level, when it comes to having like a biological machine. Like for instance, like Dynein motors. Do you know what a Dynein motor is?
Is that the one that's in the ATP synthase or the flagellar modem? I don't remember which one is which. Sorry.
So
we are familiar with the irreducible complexity stuff. We have
there is that, because that is to sum up that is basically the argument from Darwin's Black Box by Dr. Michael B. Heath, correct?
Yes.
Okay.
That's very, very similar to that argument. That's actually kind of where the idea started, and it's been kind of built upon since then. Yeah.
Okay. So I want to make sure that I'm clear about what the claim is that you are making with this, because I've seen a few different versions of the irreducible complexity claim, and I don't want to try to talk about one of them if it's not the one you're making. So the versions that I've heard are: one, irreducible complexity is a argument against evolution, and so two things are true. Or sorry, one of two things are true. Either irreducible complexity or irreducibly complex systems can evolve, in which case the argument from irreducible complexity falls apart. Or two, they really are genuinely impossible for evolution. That's one version of the claim I've heard. I've also heard the claim that Darwinian mechanisms, taken strictly, by which we mean heritable variation and natural selection, cannot account for irreducibly complex systems in life, but that other aspects of evolution, like neutral evolution, or say horizontal gene transfer or things like that, might be able to account for them. And so therefore, if you can demonstrate that something is irreducibly complex and is not a result of those other forces, then that might be a problem. And then the last one that I've heard, which I think is actually the one that I've heard the most frequently, is that some irreducibly complex systems cannot evolve, but others can, and we need to go on a case-by-case basis. Do any of those sound like your position? And if none of them do, what exactly is the argument that you would make with irreducible complexity? I'm sorry, I can talk, I promise.
The gist of my argument, I should say, is that there are systems that we find in like cells, for instance, that are very analogous to, say, either a computer program or like a robot, in that they are absolutely irreducibly complex to the point in which you cannot account for their creation via a step-by-step naturalistic process. And in it, like I said, you can go to the analogy of using like a computer or like a computer program, because no competent IT person would ever say that they think that you can get a computer or a computer program via random chance naturalistic processes. And so essentially that is kind of the argument, is that there are systems like those in our cells, in other animals, and all over the animal kingdom that cannot be accounted for via naturalistic chemical processes.
So I think I think that eliminates the sort of middle option that I said, where the strictly construed Darwinian mechanisms can't account for it, but other aspects of modern biology can. So you would say that there are no unguided processes that could account for irreducibly complex systems. Would that be accurate?
At least not that we know of.
Okay.
Correct. Like I
Okay.
said, it would be like a computer program. It's like there's no current mechanism that we have that would somehow explain a even if you started with just a computer chip, you're never going to get that to randomly assemble itself into a computer, basically.
Okay. And so with that, then there are the sort of the remaining two. And I think you're going for the stronger of the two, which I'm glad to hear, because it's the one that actually is an argument. Which is that if any irreducibly complex systems could be shown to evolve, it is no longer a problem for evolution that irreducibly complex systems occur. However, as far as you are concerned, there are no such systems that have been found. Is that an accurate statement of the argument?
I'd say so.
Okay, and I want to make sure that we are on the same page about irreducible complexity, because it's also something that people have gone back and forth on. And I usually take basically the definition that Behe gave in Darwin's Black Box, because I think he actually laid out a pretty good definition, and I don't have it memorized, so forgive me if I go a little bit off of his wording, but I think I have the spirit of it down pretty well, which is to say that an irreducibly complex system is a system of finely tuned parts that interact with each other such that the removal of any one part causes the function of that system to simply cease occurring. And that these parts have to be well matched and interacting, which I guess I already said. But does that seem like a reasonable definition? And so some examples might be like the human eye, or the flagellar motor, or the mammalian blood clotting cascade, where if you're missing a single part, the function is either greatly diminished or non-existent.
Yeah, that's a pretty good definition, I'd say.
Okay. All right. Well, the problem there is that, I mean, such systems have evolved under direct human observation within historical timescales. So it seems like it's not a problem for evolution, because we've seen it happen. It's happened in HIV group 1. It's happened in
subfunctionalization of any gene basically is irreducible complex, or results in irreducible complexity.
Yeah, we've also seen it evolve, irreducible complexity evolve in Escherichia, E. coli. I can never get the genus name right when I'm just trying to pronounce it. And so we also have step-by-step accounts for other irreducibly complex systems, including the mammalian eye. In fact, we've had a step-by-step account for how that works since Darwin. That's never been a mystery to evolutionary biology, even from before, the 20th century. So it seems to me like this argument simply doesn't work, because, I mean, if you want, we can go through specific examples of irreducible complexity that have been known to evolve. But
so, like in the case of the dynein arm, or the ones that you're these proteins, for instance, like the eye was considered, a really good example of this, but we have a myriad of explanations for how eyes evolved, and so that one doesn't seem to be used as much anymore. There's been a lot of research on the evolution of flagellar motors, or the blood clotting cascade. We know now that like proteins that are necessary for that exist in like echinoderms who don't have blood, so they don't do blood clotting at all. And so it's like it's kind of like whack-a-mole with these sorts of things. It's, well, we whacked this one, so now, okay, well, we found a new cellular structure that this one is definitely irreducibly complex. Okay, well, that one isn't irreducibly complex. Okay, well, this new structure is irreducibly complex. Okay, well, not that one. Okay, well, we found this other one.
Actually, Jackson, I slightly disagree with you. I actually do think those are irreducibly complex.
Oh, okay. Well, in the sense that like we can't find a like an evolutionary explanation for.
But see, that's the thing. That's why I wanted
to
try to define irreducible complexity, not in terms of evolutionary history, but in terms of current observable aspects of the system, because that way it gets rid of the question of evolution in terms of figuring out if a system is irreducibly complex. And I actually do think that many of these systems are irreducibly complex under the definition Behe gave. Like, I would not argue that the blood clotting cascade or the flagellar
You
for
have to
motor
have all the parts
physics.
for it to work.
Right.
Right. Exactly.
It meets the definition.
Right.
So sorry, that might have been a little bit inside baseball. Me disagreeing with Jackson.
No, you're right. So
I mean, I agree. I agree with you. Yeah.
can I address that then?
Absolutely.
So there are 2 points why I would say that that's not quite an accurate statement for saying that it's not a problem for evolution anymore. Because, so the first reason is that you are correct that we have observed irreducibly complex systems originate from other irreducibly complex systems that already exist. But what we have never observed is an irreducibly complex system originating from a non-irreducibly complex system, as in like dead matter arranging itself into an irreducibly complex system. So that's problem number one. And then problem number two is the information behind these mechanisms, because these types of machines, the reason why I use the analogy of a computer is because it's a very, very apt analogy, because you have hardware, which is the proteins and chemicals and such that we're talking about. But then on top of that, you have the information that codes for that. And what has never been accounted for or observed to originate is irreducibly complex information that is used for the coding / creation of these proteins that form these irreducibly complex systems that we find in the cell. And so because of those two things, it still is a major problem for at least abiogenesis as an originating factor, because there's no mechanism to account for the origination of new information from non-information, first of all. And second of all, there's not any observation that we have made from, like I said, like a dead chemical, dead chemicals
like
arranging
cells
themselves
coming from
into
non-living things. Yeah.
Yeah. And because of those two facts, it is still a problem.
So a couple of things. First, typically what Dapper and I do when we have these conversations is we usually skip abiogenesis, because we usually just talk about, like I said earlier, we're not biochem people, we're not like systems chem people. So we usually just kind of skip the abiogenesis part. We say, like, listen, if you want to say God exists and he just snapped his fingers or wiggled his nose, whatever, and the first came into existence, typically we just grant that and we say, okay, now let's talk about evolution.
Nose wiggling is for witches and warlocks, not deities. Come on.
Okay.
Get your sitcoms right.
My bad.
Actually, blinking, that's for genies.
All right. Well, so I'm okay with making that caveat, but before we make that caveat, can I at least get you to acknowledge that those two facts kind of still mean that it is a problem for evolution?
For evolution, no.
Right now?
No. And I'll explain why for the second one. So for the first one, like I said, I'm not going to argue about like systems chemistry, because I'm not a systems chemistry person. But the information one, now Dapper and I typically, we will very much disagree with that one, because, well, first we need to establish, just like we said with irreducible complexity, what do we mean when we use the term information? What are we saying information is with regard to? So if we're talking about, you mentioned proteins. So when you say, when you're talking about information in terms of proteins, are you saying like protein coding genes? Is that what you mean by information, or just genes in general? So do regulatory genes count as information in addition to protein coding genes? What do you mean when you use that term?
The information that I'm referring to is the information found within those genes. And at least specifically in this case, we're primarily talking about coding genes for proteins.
Okay.
In which it is information that it uses to code for the protein. That's kind of the whole basis of the protein synthase process.
So, for instance, I'm sorry. You go ahead, Dapper. I've been talking.
All right. So I don't know if that answers the question. Maybe if we ask it another way, we can tease out a rigorous definition for information that we could use. Let's say that I present you with two organisms, and we'll say that they're similar organisms, right? Like we'll say a tiger and a house cat. Do you have a way to determine, given however much laboratory access and resources you need, right? It doesn't have to be something you could do right now, just something hypothetically that you have a way to determine whether the house cat or the tiger has more information and how much more or less each has than the other. How would you go about doing that if that were your task?
So describe the task specifically again. What do you
All
mean?
right, so I present two relatively similar organisms, a tiger and a house cat, and I think we can agree that those are pretty similar organisms.
Yeah, they're pretty similar.
Okay, I mean they both even like boxes. So, and my question to you is: given as much lab equipment as you need and as much money as you need, could you devise an experiment or a measurement or something like that that would tell you whether the tiger or the house cat has more information, and by how much? Is that a thing that you could conceive of a way of doing? And if you can, how would you do it?
Like I said, like you said, I think that means we need to figure out what we mean by more or less information, because we could define it as like base pairs or something like that. And if you define it as base pairs, then all you have to do is just count up all of the base pairs in each genome, and whichever one is bigger technically has more information in it.
Okay. So,
but it really defines on what you mean. Yeah.
So, would you be comfortable saying that the organism with the biggest genome has the most information?
I'm not comfortable with saying that, no, because I'm not sure that that's the entirety of what we mean when we say information. I do think that that is an example that we could use to quantify information.
Sure.
But I don't think
always work.
Yeah, I think there are instances in which it wouldn't work because, like, for instance, there are like, let's just for example, like tomato plants, I believe, have more genes than humans do.
Yes, by big margin.
And I don't think any competent biologist would tell you that a tomato plant is more complex than a human body, and so
that's another thing that I don't really know how to measure. I don't know how to compare complexity in a human versus a tomato plant. Because, I mean, vascular systems, the various pores that plants use to breathe, the chloroplasts, all of that is extremely complex, and humans don't have anything like that. But on the other hand, plants don't have anything as complex as an animal brain that's controlling their action. So it's hard to measure. But so my problem now is that you've attempted to give a measurement option, which was just gross genome size, but then backed away from that because it's very obvious that many organisms which you would consider to be simpler and therefore have less information than humans have a bigger genome. For instance, the biggest genome known to science currently is that of Paris japonica, the Japanese canopy plant, which is just this little weird shrub thing that has a flower in the middle of it.
You're right.
So if it's not genome size, is there a way that we could reliably compare any two organisms and come up with a number and say this number is higher than that number, therefore this organism has more information than that organism?
I think we could quantify the level of complexity of each organism and go from there. And you can define what type of information you're talking about from there. Because when we're talking about when something is complex, and again, I don't think there are a whole lot of biologists that would disagree with me here, is that when we mean complex, we just mean like has a lot of parts that are intricately interwoven that all function simultaneously. That's what we mean by complexity.
I mean, that's subjective though.
No, that's pretty
No, it
objective.
is subjective. There's nothing objective about that definition. Just saying, well, there are lots of parts that are complex and that work together.
Let me
well,
give
I
an
don't know.
example
Okay.
of an organism that has far more parts than you do, but I think would probably be claimed by you to be more simple. And that would be clonal forests, right? A clonal forest is a set of trees that are all actually the same organism, and they have an interconnected root system. But when you count up their parts, it quickly goes into numbers that are hard to name because every single trunk has its own trunk. It has its own vascular system. It has its own system of releasing sap versus what's that other thing? The thing that
Right.
amber comes out of resin.
Okay,
Resin
so I
has
would
to
say
release-
that that's kind of a false comparison, though, because overall the organism is made of the same general parts. Like each tree individually has overall the same general parts compared to each tree, and that's kind of what we mean. Like if you go off of just the basic definition of complex, it is a whole comprised of interconnected or interwoven parts.
Mhm.
And so in order to be more complex, all you need is a whole that is comprised of more interconnected, interwoven parts.
What counts as a
And
part, though? Is it
I
each
mean,
cell?
most of
and
your
hold on,
cells are
and
made of the same parts too.
hold on, and what we refer to when we're talking about this with biological complexity is individual parts, as in humans overall have more individual parts than any a big large biomass of trees, and that's because each tree individually doesn't have different individual parts. Overall, the tree itself has the same general individual parts.
Well,
And
now the
so
question is, what is an individual part? Is it a tissue type?
like an individual unique part, something that's not the same as the rest of the parts.
That doesn't
Like,
help me.
so
Is there a
if
way
you
can
have
define it
so
without
if you
using
have
the word
two of
part?
the same part, that doesn't mean that you have two parts.
Okay. How many parts are in a human hand?
I don't know off the top of my head, but you could
How many parts are
absolutely
in a human
quantify it.
tooth?
Again, I don't know off the top of my head, but you could quantify it.
Is there any part of the human body for which you could list a number of parts that is larger than a cell?
Yes, absolutely. What?
Which one?
All of them, especially like the human brain.
Okay, how many parts are in the human brain?
Again, I don't know off the top of my head, but you
Well,
could
that was the
absolutely
question. Are there
quantify it.
any parts of the human body for which you, Terry, can actually tell me how many parts exist in that aspect of the human body? Is there any particular anatomical feature anywhere in the human body that is larger than a single cell where you, Terry, can give me a number that is the number of parts in that thing, whether it be a hand, a toenail, a tooth, your lacrimal bone? I don't care. Anything that you can name and then give me a number for the number of parts in that
Not
thing.
at this time, if that's what you mean.
Okay. Is because I've never seen anyone give any such kind of accounting. And that's one of the reasons why I think this is probably subjective, because it just depends on how granular do you want to be with your description. Like I could say that the parietal bone is one part, right? But Jackson could come by and say that every single squiggle in the suture of my parietal bone is actually its own part because each suture is unique, and each squiggle in each suture is unique. And therefore my parietal bone actually has hundreds of parts, right? Or you
Correct.
could say that actually
Okay,
the parietal bone also has osteocytes and osteoblasts, and those
Alright.
are their own separate parts in addition to every single suture.
You are correct that if you use that basis, that it can be subjective. But what we are talking about is the biological context of protein synthesis, as in how many proteins slash genes slash interwoven parts are in the animal. It's not just one factor.
Okay.
That's kind of the whole point, is that the definition of complex is just interconnected or interwoven parts.
Okay, but then
And so the
are-
more of those interconnected and interwoven parts you have, the more complex you are. And so the fact that we can go all the way down to a point in which a human is more complex than, say, a tomato plant, then that is at least evidence to suggest that a human is more complex than a tomato plant.
Okay, but there's the problem, right? Because if the number of proteins and the number of genes also factor into this, then I would argue there is no objective way to say that tomato plants are less complex than humans. In fact, I would say that if that is what you want to go with, the number of genes and proteins, then in fact, tomato plants, along with their relatives, the grapes and potatoes, who all share this ancestral whole genome triplication, are all more complex than humans on the basis of that measurement. But you've already said that that's not okay with you. So what I'm keep getting from you, and I'm sure it's not intentional, but what I perceive is that you keep telling me that complexity is this, and then when I try to actually turn that into something that is objectively definable, that we could use in science as a measurement, that we could learn from, it ends up contradicting something else that you believe, and so you have to go back and modify that. And in fact, at this point, it seems like you've gone back to a previous definition that you already rejected because it did lead to
That's
things
only because you
being
keep
more complex.
presenting these positions that are kind of forcing me into that manner, because you're
Yes,
asking me
that is
to.
my goal.
Yeah, you're right, but that isn't an actual requirement, is kind of my point.
It
Is
is
Well,
in
you want
science.
it to be logically consistent, right? You want your position to be logically consistent such that all your
correct, and
definitions are not like contradictory
I've
to each
already
other.
shown you that you can choose basically whatever definition you want, and there are many different definitions you can choose if you're trying to define uh, the amount of information within an
So
organism.
may I?
But if we're talking about the overall complexity of the organism, as in all of the parts and how many there are, how many they're interwoven, etc.,
Hmm.
that's where we're talking about the complexity of an organism.
So, if I may, these terms, for instance, like information and complexity, are not really ever used in these contexts in biology in the way that you, as well as other people we tend to talk to, use them. Like scientists don't really go around, or biologists, I guess, geneticists don't ever really go around trying to figure out how much information is in a genome, which is why the concept is really something that was invented outside of the field of biology. I had to go. I paused my screen for a moment because I had to run and grab a book because I thought of something. And same for complexity. I mean, like in biology, this is kind of a non-issue, but I was wondering if, like, I thought about cell morphotype number, like, so the different number of cell types that are in the body. If maybe like that, you could theoretically use that as like complexity as or as a proxy for complexity, for instance, in which you could say that like chordates, which includes us, have the largest number of cell types, but there's also a huge gradient in terms of cell types, right? So like echinoderms, who are the next closest relatives of chordates, have the next highest number of cell morphotypes relative to us, which is kind of what we expect if
Jackson,
evolution is true.
So,
did I just hear acorn worm erasure?
Uh, Actually, did you? Let me see.
Yes, I did.
I see echinoderms. Let's see. I'm trying to look. I see enteroprocta.
Pretty sure hemichordates are closer than echinoderms to chordates.
Hemichordates? No, they're equidistant because
Wait,
they're
related to each
ambuliparians.
other? F. You're right.
Actually,
Never
no.
mind.
Okay, so hemichordates actually have fewer cell morphotypes than echinoderms do. So sounds like those acorn worms are getting erasured. Sucks to suck, losers.
Get wrecked, acorn worms.
Anyway, back to the point though. So we're trying to come up with. You're right. These terms are we're trying to come up with a term to encapsulate an idea, right? It's kind of like what we do with species. Like, it doesn't really matter what we call two populations. Are they the same species? Are they different species? Whatever. The fact of the matter is we just need a term that we can apply consistently or relatively consistently to give us an answer, right? And so that's what we're trying to get, is an answer as to, like, well, what is information? Can we define information without using other
Yeah,
like subjective terms.
So I understand your point, and
Okay.
I would still disagree that they're subjective terms, but I would just like to go back a little bit and address something that you just said
Okay.
that isn't true, is
Which
that
is
the terms like information are not used in general biology by biologists and such like that when referring to DNA.
I mean, if you
Because
read like technical literature, that's true.
I mean, that's just not true. And I have pulled up Scientific American
I mean,
right
you can
here
find
that
usages of the term
says, hold on,
information.
that says when most people think of DNA, they think of
Mhm.
life, not computers. But
Mhm.
DNA is itself a 4-letter code for passing along information about
Yes.
organisms.
Information is
DNA
a good analogy
molecules,
to use. Yes.
Scientific
But it can be.
American is
Also not a
for
technical
literature.
source.
Right.
I did say it's a technical source, and you pulled up a non-technical source. So
I mean, I just typed it into Google to
yeah, you
illustrate
can
my
find,
point.
and you can
is
find
that
genetics papers that
it's
use the word
absolutely
information.
within the standard literature.
Using the term information as an analogy, sure, you can find that absolutely all over the place. I've no issue with that. But like trying to quantify how much information is in organisms, that's a thing that I have personally never seen geneticists do, and I've read an appreciable number of papers on genetics.
As
So,
well
like,
as discuss this very topic with professional geneticists.
yeah. So like which again brings us back to the point. Like, what
Well,
do you
again,
mean when you say
I don't
information?
understand. So hold on, before you continue, I still don't understand why you're putting this arbitrary requirement on it that I must find some sort of technical paper that uses the word information. When
That was defending
when
your
I
point.
pull up Google right here, I can show you National Geographic, Nature,
Not a technical source.
MIT,
Oh,
Okay.
Britannica, BBC, Genome.gov.
What in nature? What in nature specifically?
DNA is a structure that encodes biological information.
Okay, but can you find like a technical source that defines information in a way relevant to organisms such that we can quantify it?
I'm sure I can, yes, and it might take me a little bit of time, but my point here still stands that it's obviously not something that is not used by biologists in the
No,
standard
your point does
literature.
not stand actually, because again, you're just doing the same thing over again. I said this is not a term that has a quantifiable definition that is used in the regular literature by geneticists or zoologists or paleontologists or whoever. And you said, well, I found sources that use the term information with regard to genetics. I don't dispute that. Lots of genetics paper, you'll probably even find like genetics technical literature that will refer to information in the genome, but I have literally never seen them. I've never seen them define it in a way that like either the Discovery Institute defines it or Answers in Genesis or the Institute for Creation Research, because typically when technical papers talk about it, they're referring specifically to either regulatory sequences or to protein coding sequences. And as we already established, you don't agree with using those as your definition or your examples for information, which makes it irrelevant then. So we're still stuck with how do you define information?
Yeah, but you're not entirely wrong, but to again suggest that it's not in the literature is just false. Like
Then find me an
I can't
example.
find you right now what is the standard definition of biological information unless let me just Google it right now.
So, like again, like when you may even find an example in which researchers refer to a protein coding gene as like encoding information, but and that's fine if you want to use information as like the a sequence of nucleotides that codes for a protein, right? If you want to say that or that binds to a protein, if you want to say that that's information, I'm fine with that. That's perfectly fine with me. But then we have to acknowledge that those sorts of sequences form under natural conditions, and we've witnessed that. And so, like, then we come back to okay, well, in that case, information has natural sources, which raises more issues, as I'm sure you can imagine. But like, when the Discovery Institute or when Answers in Genesis talks about, or Institute for Creation Research, whoever, when these organizations talk about information, they're never using it that way. If you do want to use information as just referring to protein coding
No,
genes
I
or
think
regulatory sequences,
so.
that's fine.
So
That's
I
what
think
we're saying.
hold on. hold on. I think that's just a fundamental misunderstanding of what's going on here. And I didn't mention this earlier because it didn't really occur to me. But that's not what we're talking about. When we're defining information, what we're talking about is data. And that is the definition of information, is that the data within the DNA molecule is the information. And I've got a thing from Stanford right here that's entirely about biological information.
With regard to protein coding genes, and regulatory sequences, I'm sure.
No, this is specifically talking about informational language that is used by DNA and RNA molecules to code for proteins in molecules.
Yep.
And set of facts of
Protein coding genes, yes.
Yeah.
I missed my step there, which
That's okay. Anyway, Dapper, do you want to add something on that?
Yeah, so
even this use of
I feel
coding
like
language is completely
my concern is
uncontroversial.
now that the word data absolutely does not apply to DNA in any way. Data is specifically information and statistics that have been gathered for analysis. Until there is a person attempting to run some kind of analysis, it is not data. Your ribosomes aren't trying to do statistics on your DNA. A scientist
Maybe
might
yours
do
aren't.
Well, a scientist might try to do statistics on your DNA, but that doesn't become data until someone is about to go do that thing, or is it collecting it for that purpose. Data does not exist objectively in nature. It is a thing that humans do with facts. They collect them as data for later analysis. So to say that the information is the data in your DNA makes as much sense as, I mean, it literally doesn't make sense in terms of how the word data is usually used. So I don't think that that's going to be a valid explanation. And so
I think that's completely false in the misrepresentation of the actual biological systems involved. I would like, you to find me a biologist or a geneticist that would tell you that it is not information that is found within DNA, first of all.
I mean,
and
I
second
know,
of all,
off the top of my head,
if you want a rigorous definition of biological information with regards to DNA and RNA systems, I found one pretty good from World Scientific. And it says, we propose that biological information includes all manifestations of information in living organisms. This description has the potential to include all categories of information. From scientific studies in genetics, it is clear that there are many subsets of biological information/codes and many more wait to be uncovered within the DNA/RNA systems alone.
That didn't tell us anything.
One, it is reasonable to believe that progress on biological information will be accelerated in each information subset. Towards this goal, we should begin by defining a definition, a description, or explanation of a word or thing by its attributes, properties, or relations that distinguishes it from all other entities. That's what I was saying when we're talking about unique individual parts. And
that didn't tell us absolutely anything.
So just parts are things that you can define?
No,
that's what that sounded like.
Defining the description or explanation of a word or thing by its attributes, properties, or relations that distinguishes it from all other entities.
Yes, that's what a definition is. So
yes, that's what it's telling you. And what I'm telling you is that that's what I was referring to earlier when I said individual unique parts. This is a much more rigorous definition of what I was saying.
Okay, so a part is anything with a definition.
No, a part is anything that has attributes, properties, or relations that distinguishes it from all other entities.
Right, but that thing that you just described there is what a definition is. So a part of an organism is any aspect of an organism that I can define. So a hand is a part, but it's also made up of the parts of each individual bone, which is its own part, because I can define those. And each ligament is a part.
Correct, and that's what it means when it says that there are different types of of biological information.
Okay, so
But
that's
this
where we do come
definition
to a
is just
problem.
trying to, well, this definition is trying to encompass all of it. This is trying to say, okay, overall, including all the genetic level and the nitty-gritty, all the way up to all the big arbitrary definitions of biological information you can make. This is what it means. And I brought this up as a point, counter to your point that this isn't used in the biological literature, and see that this isn't well defined, because it's at least well defined on this scale. And I'm trying
On
my
the
hardest
scale
to
of
find
just
a
findability?
peer-reviewed, published scientific paper that's regarding this, and I will find you one.
Now,
But this still illustrates the point that this is obviously a term that's used very, very commonly in the biological literature.
I feel like I must be misunderstanding you, because what I'm drawing from what you're saying isn't making a whole lot of sense to me. So I'm going to assume that that is me just being a big dumb guy. Um, uh, so because what I am hearing you say is that we can define a part as any aspect of an organism that can be defined, but the problem there is that that means that organisms have essentially infinite parts. Because I can dive right down into things like a part of you is like, you know, the theta wave of that one electron that's hanging out in that one hydrogen molecule, that's an aspect of like that one lipid that's somewhere in the left part of your liver, right? That's a part. But in that case, then the thing with the most parts is the thing with the most mass, right?
Which would be
That doesn't
Pando.
seem- Yeah, which would be Pando, a clonal forest. So we're right back to a clonal forest, but you've already disagreed with that. So it's, once again, we're back to a definition of information or parts
All
or
right,
complexity
so
that you. I'd like to finish my sentence, please.
Oh, okay.
That you have retreated to after being shown that the previous definition comes to conclusions that you don't like. And I guess I would just really like it if you could pick a definition and stick to it that actually, and if it turns out that it has results that you don't like, then you just accept that there are results that you don't like. Because what's happening right now, as far as I can tell, is that you've retreated to a definition that is so useless that you basically turn biology into the coastline paradox, which isn't very handy for biologists.
I would say that's a very complete misrepresentation of everything that's been going on here, because what's been going on is I've been providing you many of the different definitions that are very, very established in the modern literature for biological information, and you keep bringing up instances in which that definition of biological information doesn't apply. And what I'm trying to tell you is that they are different definitions depending on the context. And what you're trying to do is say that I'm reverting back on my definitions, which isn't true. And if you would like me to quantify it, I have pulled up yet another article from World Scientific. It's actually a paper. And in here, it says, what precisely is information? Anyone who has studied this field is aware of three working definitions of information: classical information theory, algorithmic information theory, and complex specified information theory. And these are the three main definitions of information in general when we're talking about biological systems. And the first definition, classical informational theory, is also referred to Shannon statistical information. It deals solely with the technical and engineering aspects of communication, and this involves analysis, including obtaining statistics on the materials, symbols for data transmission, storage, and processing. The second definition is algorithmic information theory, or the Solomonov definition of information, dealing with the complexity, quote unquote, as this term is defined in the theory, of material symbols in data structures and of the objects in general. The third definition, complex specified informational theory, or the Dembski informational theory, roughly the same as classical informational theory, but adding the important concept of specification, quote unquote.
Wait a sec. Wait, hold on. What is this what did you say it's called, this website?
World Scientific. It's a Singapore-based publishing house. Okay, which of these three definitions do you mean when you say that organisms can gain or lose, or hypothetically could gain or lose information in reference to irreducible complexity? Which one are you using? Because none of those are the ones that you gave previously.
No, they are basically all the same ones that I was using earlier. I just didn't specify between them. They're just much more complicatedly worded and much betterly worded because I have a hard time wording
Okay.
things.
But there's
But
still
I would
three of them.
definitely, and according to this, it seems like they're pretty interchangeable as far as the definition of information.
I'm sorry. Are you saying that Shannon information is the same as specified complexity in information?
No, I'm not saying that at all. I'm saying
well, then they're
that
not interchangeable. But
I must be
someone here,
reading what this says.
There is a gigantic miscommunication happening somewhere, and I don't know where it is, and it is frustrating because I want to understand what you're saying, and I think you want to understand what I'm saying and what Jackson is saying. But at no point does it seem like that has happened since you brought up the word information.
So I think what you're missing is you're not understanding what that there is an actual biological definition of like information found within DNA, and it's not just like coding proteins and stuff like that. It is the 4-letter alphabet that's in the code.
Okay.
That's kind of the whole point.
So the sequence is what the information is.
Pretty much, yeah. That's how it's transferred. That's how it's read. That's
but Terry,
how it's used.
Then we're back to grapes have more information than humans because they have more sequence.
Because that's not at all what we're talking about. We're just talking about the information in the DNA. We're not just talking about coding proteins.
Right, but you just said that was the sequence. If grapes have more sequence than humans, and the sequence is what the information is, then just by plain old math, this number is bigger than that number. Grapes have more
coding proteins.
Well,
what I'm talking about is everything, all of the information, not just the stuff for coding proteins.
Great. I'm also talking about all the information because grapes also have more non-coding DNA than you do. There is no way in which you can say that the sequence is fundamentally what is information, whether you restrict it to coding genes or not, in which you do not recover grapes and potatoes as having more information than any known animal. That is just a fact of math.
Except maybe lungfish.
Maybe lungfish. They're weird. But like, certainly the Japanese canopy plant beats the lungfish, so we'll go with that one. If your definition has anything to do with just the bare actual sequence of nucleotides in the DNA, then the Japanese canopy plant wins the information competition. That's just a mathematical fact. If you don't want it to be that, you have to come up with something more abstract than just the A's, C's, T's, and G's that make up the chromosomes of the organism in question. No matter how many times you say, well, that's not really what I mean, when you actually drill down into how do we quantify this and your answer comes down to nucleotides, then I'm sorry, but the Japanese canopy plant wins and you lose in the competition of who has more information, humans or Japanese canopy plants. But that doesn't seem to be an acceptable answer to you, which means you have to come up with a different definition. And just saying, well, I don't think you're understanding me, doesn't really cut it, because I'm beginning to think that the miscommunication is between the words that you're reading and your mind, and not between your mind and my mind.
No, I don't think so. And the reason why is because going back to what was said a while back, as I had said, we are talking about the overall complexity of the organism, which takes into account the biological information in addition to the overall amount of parts that constitutes the organism.
Jackson,
that's kind of the whole point.
Jackson,
is that the human brain has more moving and working parts than most plants on the planet.
But it has fewer than most cetacean brains. So what do I do with that fact? Is the beluga whale has more information than humans because its brain has more cells than your brain does, which it absolutely does.
No, not necessarily. That's kind of my whole point, is that it's not defined by just one of these aspects. It's
But it has all the
defined by
same
all of them.
parts. It has all the same parts as your brain because it's a mammalian brain. It's a eutherian brain. So it has all the same parts as your brain, and it has more cells that make up all those parts.
Jackson, have you ever been to a water park?
Yes.
You ever been on the lazy river?
Yes.
It's a good ride, isn't it?
Yeah. I mean, I guess so.
It's-
There was a
it's
really
less-
cold one at Schlitterbahn, but yeah,
Oh, it's unfortunate.
the one
It's-
at the
It's less fun as a conversational trajectory, though.
sorry. Okay. Okay. So my problem, I think, again, the problem with like appealing to complexity is we're appealing to another undefined term to support our undefined term. Like,
No,
not that we can't find definitions for it. We can find definitions for information. The issue is we're trying to figure out which definition you're using. That's been kind of the central issue. And actually, I do remember you did also say that you only had an hour. So if you want to like give your last few thoughts.
Oh no, that's all right. Sorry, I forgot to let you know that ended up clearing up. I've got
Oh,
like
okay.
another
Oh, okay.
30
Yeah,
I've got, like,
I didn't
another
want to
30
step on
minutes
your
or so.
time. You know.
No,
so-
you're fine. Thank you for
Okay.
reminding me, though. I appreciate that.
Yeah, of course.
No, hey,
we
we're
care about
here.
people's time.
Yeah, we're here to make things as pleasant as possible, not to just.
Yeah, fair enough. Well, so I think you're right that this whole confusion is due to a misunderstanding, and it's on both of our parts, because it's on my part for not understanding the scientific consensus definition of biological information. And it's the misunderstanding on your part that there is a biological consensus on the definition of biological information. And I've
not
pulled
my
it up
position.
from the Royal Society.
That's not my position.
And well, that's fine. You're allowed to disagree with me if you want.
I mean,
but,
my position is I don't care what the definition is, so long as we have a consistent and useful definition. That's why I
all
made
right. Well, here is
reference
one
to
that I
species.
think I'm willing to commit to, because this is
Okay.
at least according to the Royal Society, this is the standard definition of biological information that we typically use when we're talking about genomes.
Cool.
It is the genotype that encodes information about the phenotype and information, or I skipped a line. Genomes are
protein
typically
coding
usually
genes.
strings of symbols, and this digital organization is naturally interpretable within the framework of standard informational theory.
Yeah.
Okay.
And so this is using the classical Shannon formula.
Yeah.
To define biological information.
But Terry, grapes have more Shannon information in their genome than you do, even if we restrict it to those parts of the genome that encode aspects of the phenotype.
And at this point, I definitely haven't done enough research to debunk that point. But that wasn't my point when I came in here. The point is
You're right, it wasn't.
The point is that they are both complex in the first place.
Okay, but I want to make sure that we're clear on why this became an issue, right? Because when you were trying to talk about irreducible complexity, one of the things that you talked about in that was information. And so in order to talk about irreducible complexity in terms of information, we had to drill down into what you mean by information in the first place. Right? Because if I say, I'm here to talk to you about piano maintenance, and I tell you that the most important thing about piano maintenance is that you always remember to have sufficient latitude, you might want to know what latitude is, because otherwise whatever I'm going to say isn't going to make any sense, and you won't be able to parse anything that I'm saying, right? Now, granted, that's just a made-up word, right? But in any context, any word that is ill-defined needs to be defined in order for the conversation to become meaningful. That is why we went to this.
Yeah,
but now that we're down to information can be said to be the Shannon information of a genome restricted to those parts of the genome that actually encode aspects that are phenotypically relevant, which means that we can get rid of the Shannon information in, say, junk DNA. Okay, great. That is a definition we can use. Now, it does lead to implications that you seem uncomfortable with, but still, I'm happy to use that definition as long as you're happy to use it with the understanding that it may result in
I think
conclusions you don't
yeah,
like.
so I think you brought up the reason why I'm uncomfortable with using it is because of that fact that the idea that there's a whole bunch of junk DNA is just not true anymore.
That's false.
That
That
is
has been
Actually,
very
so actually, it's funny you say that because one of our friends got the Discovery Institute to change their position on junk DNA in a recent debate. So it's funny you mentioned that because the Discovery Institute has liked to say, and their affiliates, like Long Story Short and Evolution News, that with ENCODE, which claimed a 84% of
roughly 84, yeah.
that 84% of our genome was functional. Again, another issue, but regardless, that they were kind of pushing that as, oh, hey, 84% of the genome is functional according to biologists. And then Casey Luskin, who is a geologist who works at the Discovery Institute, had a debate with our friend Dan Stern Cardinal, who is a geneticist, not a geologist. He actually works in the field of genetics. And he got Casey Luskin to say on camera, basically, he never committed to a number, and a part of the DNA that he would accept as having known function was less than 50%. And I believe Jonathan McClatchy followed up with a posting that said it was significantly less than 50%, because the only thing they barely agreed on, 20%, we actually have, if we are extraordinarily generous, way over generous, 20% of the genome.
But I also want to point out that for well over 50% of the genome, we have positive evidence for lack of function. It's not just that we don't have positive evidence for function. We have positive evidence for lack of function, right? We've done things like knockout tests. We've checked to see if things are under purifying selection, things like this, right? Where we actually can say, we know with high degrees of certainty that this part of the genome lacks a biological function because it can both be deleted without harm and its sequence is completely unconstrained, which means it can say literally anything in there, and that does not cause a problem for the organism that has that variant in that part of the genome, which means those parts do not function because they do not code for anything that actually is important to the organism. Because if they did, knocking them out or completely changing them would have effects, and they do not. So it's not just that we don't have evidence. We have evidence against.
So, I don't think that's true anymore. And I think it's and the reason why you say that is probably because you haven't heard of the ENCODE project. And
I just mentioned it. I literally just said it 2 minutes ago.
Terry,
well, I was talking about Dapper, yeah.
Yeah, we both heard about it. We're both familiar with the ENCODE project.
Yes, I
promise. But I must have missed what you heard, what you said, or what about it.
The ENCODE project established the what it called the functional fraction of the genome, which engaged in which it claimed engaged in biochemical functionality,
which boiled down to any biochemical activity at all.
Which technically it should have been 100% of the genome, because 100% of your genome interacts with DNA polymerase when it is getting replicated.
True.
So technically, 100% of your genome is functional.
By that definition.
Yes, by that definition, 100% is. However, when they used it, and actually it kind of flip-flopped in the there was a Grauer paper that came out that criticized them for their definition of functionality, that talked about, well, how do you differentiate if you have a random sequence of 6 nucleotides, what are the odds that they are going to be a binding site for proteins, but have spurious but it's spurious binding? And it turns out that does happen. You can find those sites. There is no protein that is produced, and there's nothing that's regulated. When the protein, like an enhancer or whatever, binds to the site, nothing happens. Or sorry, like a repressor, or nothing happens. So that happens yeah, it's there have been so many criticisms of ENCODE that the ENCODE like project themselves walked back their main conclusion from that paper. And again, Dan did a video about this in which he pointed out that the ENCODE people themselves said, like, yeah, we way overestimated what is actually meant
And,
by
and I want to point
biochemical
out that they
functionality.
walked this back a decade ago.
Yes.
This was not a recent development. ENCODE
Well,
in 2012 said, hey, roughly 85% or so functionality by our definition. 2 years later, they had a new
I would
paper
love for
giving a
you
new
to
definition.
find me that and
It's
show
ENCODE
me where
2014.
I will go and watch that video you're referring to if you would like.
Sure.
But as it stands, I am researching it right now and I cannot find it. But
Hold
what
on,
I
I'll
do
find you the
find,
paper.
yeah, send it to me if you would like. But right here on Smithsonian Magazine,
Also not
it's
a technical source.
Well, it doesn't necessarily have to be the technical source itself. And I can find it if you would like,
But go
because
ahead.
it specifically quotes the source material. But it's on your note about their definition of functional, and that's just it's a misrepresentation of it, because according to their own study, listen to this. Do, 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 do. Word functional.
Hold on,
I lost my place.
I can actually where is it? Where is that source? Hold on. Give me
in
one second. I
ENCODE.
apologize.
No, you're fine. Here, can listen to this while you're looking.
Okay.
In ENCODE, we define our criteria as, quote, specific biochemical activity, end quote.
Yes.
For example, an assay that identifies a series of bases. This is not the entire genome. Parentheses. So, for example, things like, quote, having a phosphodiester bond would not qualify. When we then subset this into different classes of assay in decreasing order of coverage. These are RNA histones modifications, narrow histones modifications, DNA-acyl hypersensitive sites, transcription factors, chip sequence peaks, and D-nasal footprints, transcription factor bound motifs, and finally exons.
Yeah, transcription. Yes, that's exactly what I said. Like transcription factor. Yeah, okay. That I did mention that. So
Terry,
I said, yeah, places where transcription factors bind. Like that's what I mentioned. I did, in fact, that was the exact example I gave.
right, that's the 2012 definition that ENCODE themselves 2 years later, a decade ago.
Right, but the idea that it's
the people at ENCODE who wrote the paper that you're relying on would disagree with you.
I'm not disputing that fact, but the idea that it's not well defined so much so that you could arbitrarily define it as the entire genome is just not correct.
That's not arbitrary,
they themselves address that.
I mean, that's well, for one thing, that's not arbitrary, but they did also give if you define it as, yes, having a biochemical functionality, that if you're going to toss out the hold on the immortality of television sets function in the human genome according to the evolutionary gospel of ENCODE. Hold on. That's the paper.
It's quite a paper title.
That's Dan Grauer. That's 2013. So that's the paper where he criticized. That is the paper where he criticized ENCODE's definition of functionality. Here we go. Sorry, let me pull it up. Sorry, I don't usually have to run down sources during these things.
Jackson, while you're working on that, I have a quick question, or I guess
I
found comment for Terry while you're trying to pull that up. So, Terry, one of the things that concerns me a little bit about how this is going is that you just pulled up a definition for function that was the one used in the 2012 ENCODE paper, which both Jackson and I had already described and had been critiquing. But then you pulled up that definition as if the definition that we were already critiquing would itself overcome that critique.
Because I have yet to see the evidence that you're
Kellis
suggesting.
et al. 2014. Okay, that's the paper. That's the ENCODE paper that walked back their own definition.
What is it?
Kellis et
I'm sorry?
al. 2014. Hold on. Defining functional DNA elements in the human genome. Yeah, here we go. So this is, yeah, as Dapper said, this was a decade ago. Here we go. Yeah. With the completion of the human genome sequence, attention turned to identifying and annotating its functional DNA elements. This is ENCODE. This is ENCODE. As a complement to genetic and comparative genomic approaches, the Encyclopedia of DNA Element Projects was launched to contribute maps of RNA transcripts, transcriptional, I can talk to Dapper, regulator binding sites and chromatin states in many cell types. The resulting genome-wide data reveal sites of biochemical activity with high positional resolution and cell type specificity that facilitate studies of gene regulation and interpretation of non-coding variants associated with human disease. However, the biochemically active regions cover a much larger fraction of the genome than do evolutionarily conserved regions, raising the question of whether non-conserved but biochemically active regions are truly functional. Here we review the strengths and limitations of biochemical evolutionary genetic approaches for defining functional DNA segments, potential sources for the observed differences in estimated genomic coverage, and the biological implications of these discrepancies. So Kellis, yeah, so this is the paper where ENCODE themselves walked back their primary claims that 80% of the gene or 84% of the genome is biochemically functional. So there's that one. And again, as Dapper said,
but
that was a
should
decade
keep
ago.
reading. You should hold on. You should keep reading because the last sentence there it reinforces my point.
No, it
Our
doesn't.
results reinforce the principle that each approach provides complementary information and that we need to use combinations of all three to elucidate
Yes.
genome function in the human biology.
So that's
and disease.
true, and by that agreeing with that, we do not get to 84% or even 100%, whatever, of the genome is actually like either coding for proteins or is regulating sequences. That's
Correct.
still true.
Correct. I'm not making a specific
just-
percentage claim about how much of the junk DNA is actually functional. But what I am telling you is that the idea that 98% of our genome is non-functional is not true.
No
And
one's
it's
saying that
been
it is.
disproven.
Okay,
Harry,
first of
Even
all,
if it's only,
so
even
you're
if it's only 20%
Who was saying
more functional.
who was saying 2%?
so you're kind of conflicting. You're making conflicting claims here. So the 2% is I think protein coding genes.
Mhm.
It's 2%. That's not all of the sequences that are actually functional in the sense of involved in generating your phenotype. That is the 2% is just protein coding regions. There is another
Yeah,
portion that is also
you're not wrong.
regulatory sequences, which are not coding, but are also involved.
Promoters, suppressors,
Right,
things like
but are
that.
also involved in the generation of your phenotype. So that is in addition to that. So it's not just 2%. But also the as Dapper was saying earlier, we were saying that the junk DNA, we do have junk DNA. Well, you said earlier, or well, I don't want to misinterpret what you were saying. You said earlier, what was your comment about junk DNA earlier? You said like it was mostly overturned or something like that. I don't want to misinterpret what you said or misrepresent you. Terry,
you still
hello?
there? Did we lose Terry, Peter?
Hello?
No.
Terry, can you hear us?
Terry's still here.
Oh,
Hello. Hello.
Terry,
Hello.
did you have some mic problem?
Can
I
you
can
hear
hear you
me?
now. Yep.
Yeah, we can hear you now.
Sorry about
what was
that.
your,
My
what was your
internet
comment
gave out.
earlier?
Oh,
that
that
sucks.
Yeah, that happens.
That's all right. I just got my hotspot connected, so we're good.
Oh, okay.
I gotcha.
Could be worse. You
Tell-
could be
me-
losing power entirely, and that would be the end of the show if
I
you're
know,
me.
yeah.
Terry, tell your ISP that a big dinosaur is now officially angry with them, and they better get your internet back.
Yeah, no kidding.
Okay, so Jackson, you might want to re-ask that question.
Okay,
I don't
so,
think I heard it.
yeah,
um,
you need to rehash.
Okay,
whatever you
so
said.
that's fair. So well, for one thing, it's not just 2%, right? Because regulatory sequences, while they don't code for proteins, are involved in the generation of your phenotype. So I was asking what your comment earlier was about junk DNA, because you said something to the effect of that's been largely overturned, and so I don't want to misrepresent you. So could you please restate to me what you had said earlier?
Perhaps that is roughly what I said, but perhaps I should have been a bit more specific.
Okay.
That's more of what I meant, is that the I can't quite remember what you said right before it, but we were talking about things that apply to specifically a specific portion of the genome, and if
Protein coding
yeah,
genes, like, because I was saying that the 2% you're referring to is the protein coding genes, but there's also the regulatory sequences.
right. There was something else we were talking about before that where it would I can't remember what we were saying. Shoot. But it
We're
was
talking about the definition of functionality being used by ENCODE.
uh
Is that what you're referring to?
no, it was like before that. But
Before
but
that,
anyway,
mm.
but either way, the whole point that I was trying to make is that um the amount of the genome that is functional and that is universally agreed upon of being functional
Okay.
is much larger than what it seemed like you were suggesting. And
What do you mean?
I perhaps shouldn't have used like the 98% figure, because that's a very extreme figure, but it definitely it sounded like you were trying to imply that there was only a very, very small portion of the genome that what we were talking about applied to, but and that the rest of the genome is just junk. And that's what I was saying
So
is
okay,
not quite
so
true.
I think there's kind of well, I would say it's a more like around like 90%, 90% of the genome is what we currently consider junk. Now, junk in the historical sense, because there's a lot of miscommunication, misunderstanding about that term, which is why we're going to talk about it now. Junk has never meant it is all functionless in the scientific literature. So when Susumu Ono came up with that or coined that term, whatever, it meant DNA for which there is no known function. So that did not mean DNA we absolutely know does not do anything. That is what is called was it Dr. Moran, Lawrence Moran calls that garbage DNA. But then junk DNA is sequences for which we have no known function, right? So that's what Ono was using it originally in that sense. Not, and so what is junk DNA can technically decrease, right? We can decrease the amount of junk DNA by finding out, like, okay, well, this region of the genome actually does have some function. But does that mean that all of it's going to turn out to have function? No. And I'm not saying that you think that at all. But like, that's the historical usage of that term. It goes back to like a 1972 paper, I think, was when it was first coined. But yeah, so typically around like 90% of the genome is considered junk. And we do know for a pretty large portion of that that it doesn't do anything. Because like, as Dapper mentioned, like knockout tests and things like you're either missing like exons, which are the regions that actually form the amino acid sequence, or so the sequence that doesn't get the sequence that gets spliced together, because you splice out the introns and then you splice the exons together, and that's the part that actually gets read.
So
there are sequences that have no open reading frame, which means they physically cannot be transcribed.
Right, there's no promoter, etc. Yeah. So yeah, they just can't be read.
So I would like to at least understand then,
okay,
what why is that the explain to me again why that ENCODE definition was wrong again.
Well, I mean, the most basic reason is because ENCODE can't even support it. There's that. That's the most basic one, right?
Right, and that's, you want to cite that link there in the side.
Yeah, if you want to cite ENCODE as saying this is what the definition is, they don't say that that's the definition. They did, but they don't anymore. But I'll make an analogy, right? Suppose I assume you've got like a grandma or a mom or something, right?
Yeah.
Do do either of them collect like little tiny porcelain figurines and put them on a shelf like my mom did?
My grandmother does.
There you go. Yeah. Okay. So let's say, and I'm sorry for this analogy, but let's say that hopefully in the far, far future, your grandmother passes away and you inherit things from her, which includes these knickknacks. And for some reason, you feel like you don't want to throw them away because they have sentimental value or whatever. So you put them somewhere and you don't ever really look at them. But they're collecting dust. Could you say that those figurines are functional and that their function is as a place where you put your dust?
You're gonna have to repeat that. I don't think I understood what you meant.
Okay, so you inherit these figurines from your grandmother, who dies hopefully again far in the future.
Okay.
And you don't throw them out because they have some kind of sentimental value. You feel bad throwing out your grandma's old figurines. So you just put them somewhere where you don't ever really look at them and they're not in the way, right?
Mhm.
And now they start to collect dust. Is it reasonable to say that the function of those figurines is that it's where you put your dust? That they're functional for that reason?
As in like they're good at collecting dust or something?
No. The function that you need them for. This is a function for you. The function that these have in your life is as a place for dust. That's important to you. That's a thing that is a function that is helpful to your life. Would that be a reasonable conclusion to make?
Probably not.
But that's the thing. That's the kind of definition that ENCODE is going with. They're saying, hey, this particular section of the genome was just methylated. I guess its function is as a place to store methyl functional groups. But the thing is that when DNA is methylated, it makes it harder to express in the first place, even if it could be, which means it's not really important. That organism isn't getting anything out of that methylated group of DNA. It's certainly not storing methyl groups there. Methyl groups are a dime a dozen, right? It doesn't need to store them in its genome or in its chromosomes more specifically. And that's the kind of quote unquote function that we're talking about, is the same kind of function that those figurines have in collecting dust. That you don't need a place to store your dust, right? That's not a thing you need. If they were to disappear, you might not notice for days because they're not functional to your life. They're a leftover from a previous part of your life that had some function, because obviously, I'm just going to assume that you and your grandmother have a relationship that's important to you in some way. And because of that, they had a function, but now they're just collecting dust, and dust isn't a function. That's the same thing with the ENCODE problem, right? They're saying, well, this does a thing, but the thing that it does doesn't matter.
Yeah,
And so
in that
that's why-
case, being methylated or binding to a transcription, transcription factor that isn't involved in either repression or expression, that's considered biochemical functionality.
Yeah, so that's why it's not a good definition, is because it encodes completely pointless stuff as function in the genome, right? Stuff that doesn't matter if it happens or not. Stuff the cell would be just fine with or without. And that's why in 2014, those same people who made, well, I mean, not exactly the same group of people, but by and large, a very large overlapping group of people, the people who were still at ENCODE or were now at ENCODE, in 2014, walked back that estimate and walked back their definition because they realized that it wasn't a good idea to say that the function of porcelain figurines is to collect dust, because that doesn't make sense.
All right, that makes a bit of sense, I suppose.
Okay.
The only question that I have with regard to that, though, is that obviously I haven't had time to read this paper, but I still don't understand where they're maybe they are walking back their definition of what that is, but it doesn't seem like at least the scientific community as a whole has walked back the idea that junk DNA is the majority of the human genome.
Well, the only
junk
scientists that
DNA
I'm aware of who are disputing that, okay, let me rephrase. The only people even purporting to be scientists that I'm aware of who are attempting to push the idea that junk DNA does not constitute the majority of the genome of Homo sapiens are all people trying to push intelligent design and similar ideas, and who do not publish those ideas by and large in actual peer-reviewed journals. As far as I can tell, there is nothing in the actual peer-reviewed literature of science that is pushing back against this. So if
All
you
right.
want,
Well, I would just like to ask you also then,
Mhm.
according to this and according to you, would you say that transcription would constitute a function?
not
Not
unless that's actually
necessarily.
right.
Spurious
Transcription,
transcription happens.
right? For protein coding regions, or for something to be protein coding, it needs to be transcribed, right? So for some kinds of function, transcription is necessary. For other kinds, it's not, right? A promoter region doesn't need to turn into a protein, so it doesn't need to go through the whole transcription thing. Just transcription on its own is not necessary because you can think of transcription, and I'll go with an analogy because that seemed to work last time. Think of okay, you might be young for this, but you know what fax machines are, right?
Of course.
Okay. So if you've ever worked at an office with a fax machine, sometimes worthless faxes come through, right? Like, oh, hey, look, the menu for a restaurant that doesn't even deliver to the office just came through because they spammed it to a bunch of business numbers.
Mm-hmm.
So you pick up that fax and you toss it in the trash. But sometimes, like a new contract form comes through from a new customer, and you have to go and make sure that you file that in the right place and everything, right? Transcription is like whatever comes out of the fax machine, right? The cell has taken some DNA and turned it into some RNA, but now the cell is like, well, okay, I'm not going to do anything with this tiny little bit of transcribed RNA. It's not going to turn into anything. And even if it does turn into anything, it doesn't end up getting used for anything. But on the other hand, sometimes that transcription turns out a really important thing. And so it is now important. And so just like your business could do just fine without that menu from a delivery place that doesn't even deliver to your office, cells can do just fine without many of the parts of their DNA that are transcribed. And those are called spurious transcriptions. There's transcriptions that don't end up actually affecting the cell other than the energy cost of doing the transcription, which is still analogous even to the electric bill going up a tiny little bit when you get that useless fax, right? So just transcription isn't sufficient to actually say that this is function. Which is one of the things that in 2014, ENCODE walked back on, was just saying that transcription on its own is a sign of function. They said, nope, that is not something that we're going to accept anymore.
I can actually read. So this is from the ENCODE 2014 paper, referring to the definition that you gave earlier. These assays identify genomic sequences, etc. An advantage of such functional genomics evidence is that it reveals the biochemical processes involved at each site in a given cell type and activity state. However, biochemical signatures are often a consequence of function rather than causal. They are also not always deterministic evidence of function, but can occur stochastically. So that's what...
Stochastically means more or less randomly, with no particular cause or function associated with it.
Okay. Yeah, I'm just reading. If you're at all interested, I'm just reading the surprisingly the Wikipedia article on this, and listen to this. Contributing to the debate that there is no consensus on what constitutes a functional element in the genome, since geneticists, evolutionary biologists, and molecular biologists employ different approaches and definitions of, quote, function, often with a lack of clarity of what they mean in the literature. And due to the ambiguity in the terminology, there are different schools of thought over this matter.
Sure.
However,
Yeah.
widespread transcription and splicing in the human genome has been discussed as another indicator of genetic function, in addition to genomic conservation, which may miss poorly conserved functional genetic sequences, and much of the apparent DNA junk is involved in epigenetic regulation and appears to be necessary for the development of the complex organisms.
I mean, I don't disagree with most of that. I mean, and again, that is exactly what's described in the Kellis 2014 paper,
Right.
the ENCODE 2014 paper. Like that's what they're talking about is there are different methods. And again, this kind of goes back to what we were saying earlier, right? Like you can have different definitions. The issue is just pick one and use it consistently. And so what the ENCODE 2014 paper is saying is these are different definitions you can use. And if you apply it, what are the results that you get? So if you are just using like the biochemical functionality, for instance, in the sense of like if you get a transcript, does that count as functional? Well, if that's your definition, sure. But then if that's your definition, then the implication is anytime you have a transcript, even if it does absolutely nothing at all in your genome, or sorry, in your cell, then that's still technically functional, which I
Right.
mean, okay, well, that's by the definition, it's functional. But then like an evolutionary biologist would look at that and would look at the genome and say, okay, well, the regions that are conserved, so that are subject to selection, are the ones that are most likely to be functional, right? So that's the definition they're going with. And so, yeah, you
All
can
right.
pick different definitions.
Yeah.
It's just
Well,
being consistent.
And I want to
Yeah.
give. I feel like my analogies have been working reasonably well, so I want to try and give another one, right? So it is possible for multiple people to disagree on the definition of X, but to all agree that something does not meet any of their definitions and therefore is not that thing. And the example I want to use is something from linguistics, right? You're aware that Arabic is a language, I'm assuming.
Yes.
But you're also kind of wrong about that. Because it turns out that there are well over a dozen varieties of Arabic that aren't mutually intelligible. So, for instance, if you take someone from Morocco and ask them to have a conversation with someone from Iraq in Arabic, they can't, because their Arabics essentially aren't the same Arabic. And so you could say that everyone from Malta to Morocco to Iraq to Saudi Arabia to Yemen to Egypt has a different definition of what Arabic is. But they can all agree that what we're doing right now is not speaking Arabic. So even if we can't agree on what Arabic is, we can certainly all agree that English isn't it. And so even though there might be different fields in which the exact definition of function varies, all of them can agree that having some random part of your chromosome methylated doesn't indicate function. And all of them can agree that if this particular sequence never turns into a protein product and is only occasionally transcribed spuriously, that it does not have a function, even though there are edge cases where some people might disagree. And none of the definitions for function that are currently operational in biology can get us to even close to 80% function. And remember, I said currently operational in biology, which means not the 2012 definition, because not even ENCODE is using that anymore. So yeah, sure, there's differences in the definition, but none of them get to the place where we can say that junk DNA has been largely overturned. That's not an option that's available to us.
I understand your analogy and it makes sense, but I just don't agree. Just because of what I just read, and there are 3 other citations right behind this sentence that I just read, and it
What's the
says
Wikipedia
again.
page?
And much of the apparent junk DNA is involved in epigenetic regulation and appears to be necessary for the development of complex organisms.
Well, that is just wrong. Like that can't be. So if a region is involved in development, it is definitionally not junk DNA, right? Because we have a function for it. So it's no longer, we don't know what the function is. It's kind of like there's no point in explaining that. It'll be too complicated. We have a function. This is a book from, okay, I can't really access that. Okay. That is that's a paper by Matic. Oh, that's the dude who is like the genome's 100% functional. So 2 of these are books that I can't access. Non-coding RNAs and epigenetic regulation of gene expression. Like, yeah, non-coding RNAs exist and epigenetic regulation happens, but okay, so those aren't technical sources. The only one that is a technical source is this paper by Matic and Dinger. And I would be very curious, as they pointed out, as our friends in the live chat pointed out, yeah, Matic is one of the few guys who's like, yeah, DNA is like 100% functional. I would be, but yeah, I'm curious. Recently, articles have been published disputing the main finding of ENCODE of the ENCODE project that the majority of the human genome exhibits biochemical indices of function. It's primarily on low sequence conservation, the existence of larger genomes, some ostensibly simpler organisms, the C-value enigma, indicating the likely presence of significant amounts of junk. His conservation is a relative measure based on circular assumptions of non-functionality. Yeah, I'd be very curious. I'd be curious to see how this paper has been received more recently, because he's, I don't know, the arguments that he's not usually taken very seriously on this stuff, but I will. Divided by 41. Functional long coding RNAs evolved from, well, they have evolving from junk transcripts. Anyway,
Well, that is an important thing. Is that,
That is
one
true,
thing
yeah.
that junk DNA can do that is not?
oh, actually, they
technically
cited that
function.
paper.
So one thing junk DNA can do that is not technically function is that as a result of mutation, it can transfer from being junk DNA to
Yes.
being functional. Now we see
Right.
that happen all the time. Well, I mean, I don't know about all the time, but like that's the main way that we get so-called orphan genes, right? Like taxonomically restricted genes.
Mm.
So, for instance, there are genes that say gorillas have that no other apes have, and there are genes that all apes have, but are all great apes have, but gibbons don't have, or that any other mammals don't have, and we call those orphan genes. And the main way that those come about is by what's called the neo-functionalization of previously non-functional gene or non-functional regions of the genome. Sorry. So basically, some part of the genome that doesn't even currently have like an open reading frame gains an open reading frame as a result of mutation. So it gains like a start and a stop codon, and it can be successfully read as a transcribe that can turn into a protein. And then that protein, by chance, which actually turns out the functional fraction of protein space is actually pretty high. So random proteins tend to have biochemical activity. And then if that biochemical activity has a fitness effect, then natural selection can take over. So it is true that you could say that junk DNA has, quote unquote, the function of allowing evolution to occur more easily. But I don't know that that's supportive of your broad ideas. So I don't know if that matters much.
Fair enough. But and so two things. One, I will definitely read into your paper here about the ENCODE project and how they walked that back and everything.
Okay.
Just so that we can maybe go back to like some of the main topics we were talking about.
But that's part of the problem is every time we try to get to the main topic, we end up finding that we don't agree about the very basic definitions of what's going on. So until we can agree on the basics, it's hard to get there. And as one last point on
that there's not really a good agreed upon definition that everyone agrees upon.
But we don't need that, right? Because remember, with the Arabic thing, we don't need to all agree exactly on what Arabic is in order to say that English isn't it. Similarly, we don't need to have 100% agreement on what function is in order to say that the vast majority of the genome does not have function. In fact, Jonathan McClatchy, whom you might be familiar with, he's an intelligent design advocate over at the Discovery Institute, if I remember correctly, has said, and this is a quote here, "We currently know of specific functions for significantly less than half of the genome," which means you cannot say that junk DNA has been largely overturned. At best, you could say, I expect it to be largely overturned. But beyond what Jonathan McClatchy says, we have positive evidence for the lack of function of the large portions of the genome.
Can I also point out with regard to that Matic paper, the extent of functionality? So it was referred to in the ENCODE 2014 paper, and they did not, well, I mean, they didn't disagree with it. They just kind of listed it and said it has stimulated debate. I found a paper that argues a bit later. So it does cite this paper and says, in an attempt to counter the argument that sequence conservation is a prerequisite for functionality, it has been recently proposed that certain transcriptional events may serve some role in regulating cellular function, despite the fact that the sequence of the transcriptional product is unconstrained. That's referring to the Matic paper. And it cites a couple examples. It does say, so it says, indeed, this view is in line with the findings that the transcription of certain yeast genes is inhibited as a consequence of the production of cryptic, unstable transcripts originating from upstream and or downstream promoters. Other examples have linked the generation of cryptic, unstable transcripts to chromatin modifications, DNA methylation, and DNA stability. However, it remains unclear whether the majority of unstable, non-coding RNAs have any effect on DNA or chromatin, let alone contribute to the fitness of the organism. So this paper...
So it's saying the same thing that ENCODE was and walked back on it.
This paper, no. This paper is it would be more in line with the later ENCODE paper that
was
it
walking back
remains unclear. Yeah, whether the
Yeah.
majority of unstable. Yeah. So yeah, yeah.
Well,
So-
again, that's fair, and I will be able to secede that point for the time being, just because we can probably need to get back to the main topic. But yeah, again, like I just need to read more of that
Okay.
paper, I guess. Why don't you? Can you send me that one as well?
Yeah, sure.
Oh,
All right, here it is in the live chat, or sorry, our side
pretty
chat.
much
here.
anytime you want us to send some kind of paper or something, you got it, because we like collect papers to some degree, right?
No, fair enough.
Jackson and I, we have like
Yeah.
certain favorite papers that we trade around, and it's almost like trading cards. Like, oh, hey, have you read this particular paper? Yeah, well, let me send it to you. So yeah,
true.
we have a whole backlog of, not a backlog, but just a collection of papers.
I mean, I definitely have a backlog of papers I need to read.
Well, there's that. Yeah, I still haven't read the new Abelosaurid paper from South America.
Oh, okay.
I need to
I
get
heard
on
about that.
I've looked at the diagrams and the figures. Those
You're
pretty cool.
not impressed? No.
Oh, I am impressed. I'm very impressed. Are you kidding me?
Okay, all
This
right.
is an extremely complete fossil for like compared to what I've seen Belosaurs named on other cases, like that new African Abelosaurid from a few years ago.
Okay.
It was named on basically just like part of the nasal bone. I was
Oh,
like, well, I'll grant you it's definitely deserves a new genus name, but like this is sad. And then it's like you get to this one, and it's like we've got like femur material, sacrum material, cranium material, jaw material, and I'm like, oh hell yeah. Sorry, that was just me nerding out about dinosaurs for no reason.
fair.
My apologies, Terry. That was completely off topic.
No, it's all right.
So yeah, if you need to send you a paper, we can certainly do it.
We
So
got you covered.
we'll get back to irreducible complexity, right? So my contention with irreducible complexity is that the only form of the irreducible complexity argument that matters is the strong form that some systems are irreducibly complex under the definition that I think we both agreed to, that was basically Behe's definition. And the argument is such systems cannot evolve. But such systems have both been documented evolving and have been given plausible stepwise evolutionary pathways. And the reason the plausible pathway is important is because when you say X cannot happen through Y means, if I give you even a single possible means by which Y can accomplish the creation of X, I have defeated that argument, even if the process that I describe isn't the way it happened. For instance, if you say it is impossible to drive from Toronto to Mexico City,
Right.
and yet my claim is that I don't know, Pierre over here has driven from Toronto to Mexico City. You say no, that's impossible. All I have to do to show that it's possible is show any drivable path between those two cities. It doesn't matter if that's the route he took, right? So, for instance, if you say the flagellar motor, I keep saying modem. What is wrong with me today?
The modem. What's your technology background?
Seriously, I have a degree, a bachelor's in history. What the hell am I doing right now?
I don't know, man. You're on a boat, so that's technology, I guess. I don't know.
Oh, okay. Anyway, so if you say the flagellar motor, there we go, I said it, can't evolve through natural selection because of irreducible complexity, right? If I show a single possible pathway by which it could evolve, that defeats the cannot evolve argument. Now, you could argue it didn't evolve, but that's a different argument, and we would have to have a separate discussion. And so I would argue that just simply things like the sit plus line in E. coli, in the Lenski long-term E. coli experiment. I know Dr. Dan is here in the chat. His favorite example is the change in the VPU protein of HIV-1 from SIV to be able to antagonize human tetherin, which allowed HIV group 1 to actually infect humans, whereas previously its SIV version couldn't. That's an example of irreducible complexity. We have numerous potential pathways for the mammal blood clotting cascade, which is one of Behe's favorite examples. In fact, that entire thing can basically be summed up in one word: subfunctionalization. That sums up basically the entirety of the mammal blood clotting cascade. And so because we have both plausible mechanisms for many irreducibly complex systems, as well as irreducibly complex systems that we have watched evolve in real time, the bare fact that irreducibly complex systems exist is not itself a problem for evolution. That would be my take on irreducible complexity. And I would say that all other versions of that are not problems for evolution because either A, they leave out well-known mechanisms for evolution, or B, they are unfalsifiable and therefore just scientifically useless. And I'll shut up now.
Mm-hmm. All right. Well, the reason why I would disagree with that is because I don't agree that one, it's ever been observed in the manner that is required. Because as I was stated earlier, you're correct that we have observed irreducibly complex system coming from another irreducibly complex system. But what we have never observed is a non-irreducibly complex system, or like dead molecules, for instance, becoming something that is irreducibly complex.
I mean,
So if we grant like
abiogenesis,
abiogenesis,
No, what I'm talking about is these motor mechanisms. And the second point is
wait, hold on. The motor mechanisms, what does that mean? Like the flagellar motor?
Yeah, like the flagellar motor, like a dynean motor, these things that are themselves irreducibly complex.
But we know that the flagellar motor evolved from the significantly simpler type 3 excretion system, which is still used by many bacteria. Have you ever heard of, well, interestingly enough, salmonella?
Yeah, well, right.
Let me finish.
Okay.
The way salmonella infects new hosts is by use of a type 3 excretory system, which itself is the precursor, or well, the modern excretory system shares a ancestor with bacterial flagella motors, which was basically morphologically identical to that excretory system. It uses it to stab cells and then excrete proteins into those cells that cause them to basically stop preventing the infection of the host by the salmonella. Like, that's already a thing that's we already have figured that out, right? We figured out the coalescence times for these different genes. We figured out multiple plausible evolutionary pathways to get from one to the other. So unless you want to go with, well, we have to go back infinitely in time in order to explain every single precursor of every single system.
No, we're just disagreeing on those points, and you're saying them like we know them as fact when we don't actually.
No, I mean, these are the best evidence explanations for these things.
You can maybe say that it is a best explanation currently.
We don't need to know it as fact, remember? Because the claim is that it cannot evolve. And
correct, and I don't think you have. When it comes to the example you gave of the subfunctionalization of one type of bacterial mechanism to another.
Nope, that's not subfunctionalization. That would involve
that was a gene.
Functionalization.
Subfunctionalization is with regard to genes.
Right. And this particular interaction with the flagellar motor does not necessarily involve subfunctionalization.
In fact, I'm pretty sure most of the genes involved in the flagellum are like duplicates of each other, if I remember correctly.
This involves duplication and neofunctionalization.
Perhaps I used the wrong term, but the point that I'm trying to make is that you haven't actually proved that a flagellar motor came from this other motor.
Great.
And the reason why is because of two reasons. One, you haven't actually observed that happen. And two, there is genetic information that is required for a flagellar motor that the bacteria that have the other motor don't have. Okay, and you need to account for that information.
Terry, do you remember Terry, real quick, do you remember Mon Ami Pierre, who drove from Mexico City to Toronto?
Not off the top of my head, I do not.
It was Pierre, his friend. We were saying with the Toronto,
just from earlier, you mean?
Yeah, from earlier. Yeah,
right. So Mon Ami Pierre, what you're telling me is because I have not proven to you the path that he took from Mexico City to Toronto, like I didn't show you that my proposed path that went through, I don't know, like Cincinnati. Because I have not proven to you that he actually went through Cincinnati, he couldn't have made the drive at all. But that's the thing. When the claim is this cannot evolve, it doesn't matter if I can prove how it evolved. All I need to do is show just a single possible way in which it could have evolved. and the claim that it cannot have evolved is now conclusively disproven, right?
Right, but I think that's kind of the point, is that there isn't actually a specified path in that manner, because the point A in this instance is one bacterial type of motor, and then the point B is the flagellar motor. And what you haven't specified is the actual path that the person has taken. You can say that it happened because it did, but you can't account for the mechanism that happened until you do specify that path.
Okay, so is your claim that until we can draw any given irreducibly complex system in terms of a pathway all the way back to like the first life, that therefore irreducibly complex systems can't evolve? Because
No, my point is exactly what I stated, is that you can't, there isn't even the line drawn between those two steps. And
I mean,
the reason
multiple
why that's
papers
significant,
disagree with you.
well, and the reason why that's significant for your analogy, if you're going to go with your friend that made the trip across the country, is specifying
3
the
countries.
route. Well, yeah, but specifying the route at which he went could also it could have information built into it about, like, how he drove there. If did he ride a bike? Did he
Hey?
drive a car? Did he take a
That's
plane?
not super relevant, though.
Yeah, I don't understand what the point you're trying to make is. Like,
Because
the
you
whole
haven't,
point-
because you need to show one of those things in order to actually show that it happened that way.
No, I don't need to show that it happened that way. All I need to do, because if the claim is it cannot have happened, that is completely 100% defeated by any possible way in which it could have happened, whether or not that happened. Remember, in the example, or in the analogy, the claim that I'm trying to defend, or to defeat, is that mon ami Pierre could not at all have ever driven between Mexico City and Toronto. Any single path between those two cities that could be driven defeats the claim that he could not have driven. Now, if you want to say he did not drive between those places, that is a harder thing for me to disprove. But the whole point of irreducible complexity is that it's supposed to defeat the possibility of evolution per se, period, as the source of irreducible complexity.
So
Now,
I think
Also,
that's
here's
not
a
the same
paper.
thing.
I found a paper that goes through the, or that at least goes through part of the evolution of the bacterial flagellum. And I found a video that also illustrates that process. So there's actually a couple different.
But again, so what I was going to say in response to your analogy there is, I don't think it's actually an apt analogy in this instance, because a much better analogy would be, your friend started up in Canada and then miraculously ended up in Mexico in 10 hours. And we need to explain how he got from Canada to Mexico in 10 hours. And there are some theories as to how we could possibly explain that, like if like if a wormhole opened up behind him and he fell into it or something like that, then that's a possible explanation. But you still haven't proved that it actually can happen in that manner. And that's the whole point. Sorry, what were you saying?
Well, I put in our little private chat here, a paper that's titled "Stepwise Formation of the Bacterial Flagellar System." And just to quote very briefly from the abstract, based on the phylogenetic occurrence and histories of each of these proteins, we could distinguish an ancient core set of 24 structural genes that were present in the common ancestor to all bacteria. Within a genome, many of these core genes show sequence similarity only to other flagellar core genes, indicating that they were derived from one another, and the relationships among these genes suggest the probable order in which the structural components of the bacterial flagellum arose. These results show that the core components of the bacterial flagellum originated through the successive duplication and modification of a few, or perhaps even a single, precursor gene. And then I just put that in our our side chat here.
I'm trying to find.
I guess I can put it in the live
where
chat
you
too.
are. Where is it in the article?
The paper I just sent, that was from the abstract.
From the abstract.
So I just found that paper. So I don't know. I guess I can't really ask you to read it all right now because we have 5 minutes till we end the show. But
Fair enough.
that's yeah.
But based on what you just said, and that still doesn't really provide anything other than, oh well, these genes that perform similar functions in two different organisms
No,
are
often in the
kind of
same organism.
similar.
Often the same organism, and it's not just that they're similar; they're homologous. We can actually reach a coalescence time at which those two sets of genes shared a common ancestor. Terry, I think the basic problem is that your understanding of genetics is roughly equivalent to basically like a high school kid, and you are trying to you're claiming that you have enough knowledge to reasonably say that you can overturn the genetics that's not just taught at an undergrad level of college, but that is tough for PhDs to wrap their brain around. And I'm not saying that I can wrap my brain around all of it, right? Like I said,
No, I'm
he's
not
kind of
trying
a big
to
dumb guy.
upend all of genetics or anything like that. But what I am
Terry,
trying to point out,
yes, you are. Yes, you are, because all the geneticists disagree with you, right? The only geneticists who agree with you about this are the ones who don't publish their findings in real journals because they can't support their claims. People like Nathaniel Jeanson. For some reason, I have Nathaniel Jeanson and Jason Lyle crossed in my brain, and it has been that way for like half a decade, and I don't know how to fix it. And this is off topic, and I'm sorry, but it's just
one is an astronomer, one's a cell biologist.
I get it.
So
I get it. They're very
know.
different people, but for some reason, my brain is like, nope, these are the same human, and you will continue to refer to them as such. But anyway, Nathaniel Jeanson, right? The reason that he did not publish his ideas about the human lineage in like Nature or something like that, right, is because he can't. It won't stand up to scrutiny. The people you are listening to are fundamentally science larpers. They're as much a wizard as the guy throwing beanbags and shouting lightning ball is a wizard. In that they're not, but they pretend to be for fun and profit.
well, I don't understand how you can say that when I haven't even presented a single thing from something like that. And
Terry,
I mean, that's what irreducible complexity is, though.
You presented both irreducible complexity and specified information. That's
correct. And I never made the
complexity
appeal to any of these.
So
people that
irreducible
you're
complexity
talking about.
is a talking point specifically pushed by I mean, it was like the modern version of it was coined by Michael Behe, who worked for the Discovery Institute, and of course it's used not just by him, obviously it's used also by like the Answers in Genesis, the Institute for Creation Research, all these groups. But these groups are the only ones who use this terminology and try to use it to argue that evolutionary theory is false, like writ large, right? It's just them, and they don't publish papers on this in the relevant journals. They don't even try, really. And I mean, like, what do we do with that? Like, when these people do publish actual literature, like for instance Gunter Beckley, who does publish actual literature, I know because my thesis was on my thesis for my master's degree was on dung beetles, ones that are in my area. And when I was doing research, or for my background research, I came across one of his papers on the evolution of beetles, of Coleoptera, the order of beetles. Now, when he wrote that paper, there is nothing at all about irreducible complexity in that, or specified complexity, or really any even information, anything like that in that paper, because they know the topics. They know that they can't appeal to these topics because their arguments don't work, right? That's why they don't do them. That's why if they do want to talk about those topics, they do it in their own in-house journals. Like for the Discovery Institute, it's Biocomplexity. AIG has like the research journal, the Answers Research Journal.
Yeah, I don't think that's necessarily true at all.
It is. It's just a fact. Like I don't know. I mean, like do a literature search. Do a literature search.
Terry,
like
you're talking to someone.
I'm aware that there isn't a whole lot of their literature in the common literature, but that has nothing to do with their science not lining
Terry,
up with reality.
I need you to understand something. Jackson Wheat literally wrote a book that documented the degree to which these people publish and account for data in what they do publish in non-peer-reviewed areas, right? This man wrote literally the book. It's got hundreds, if not thousands of pages. I opened it up today to check for a
I think it's 500
reference.
pages.
And yeah, it's huge. I was like, oh, this is page 200 something. It's probably somewhere in the middle of the book. No, it was in like the first quarter. Like, it is, you are talking to the guy who did the work to demonstrate that no, they don't publish in real journals. No, they don't account for any more than about 10% of the data in any given case. When you say, I don't think you know, you are talking to someone who spent years of his life.
And we're working on the second one.
No.
And we're working on the second, examining and documenting and publishing on exactly the thing that you don't say, that you're saying he doesn't know about.
Like,
You
we've
didn't let
literally
me
read
finish in the
hundreds of their. Okay, go ahead. Go ahead.
The whole
Let's
point
try to
isn't-
wrap this up.
Yeah, that's fine. We can try to wrap it
Yeah,
up.
go ahead.
But the whole point, yeah, you're right. There isn't a whole lot of their publications in the normal literature. But again, that has nothing to do with the actual data that they're presenting. The data that they present is absolutely sound and
No, it isn't.
absolutely
Okay.
holds up the scrutiny.
No, it literally isn't. So, like, I again,
And I
when
would
I
love
came
to read your
across-
book. I would love to read your
The
book.
Rocks
when
Were
you
There,
want that.
Volume 1 by R.J. Downard and Jackson Wheat. I work on
It's
Volume 2.
on, it's
But
on
like,
Amazon.
yeah, we've read RJ. I don't know if RJ's still in the chat. He was earlier. Yeah, because it wasn't just me. Yeah, RJ's still there. It wasn't just me. We co-authored this book. But yeah, we went through hundreds of citations, in which we analyze creationists, what they say, or not just creationists, so young Earth creationists as well as intelligent design advocates, and even some old Earth creationists, because we'll talk about Hugh Ross and Fazal Rana sometimes. I like Fazal Rana; he's a cool guy, but there's some stuff he's
Agreed.
wrong about. It happens.
Like bestiality, that's a weird take he has.
Yeah, I don't know about that, but at any rate, so like, yeah, we have read it. Like, I've read dozens and dozens and dozens of articles by creationists over the years, including like their actual technical literature that they have gotten published in the relevant journals. Like, I've read like aside from Gunter Beckley's paper that I mentioned earlier, I've looked at like Jonathan Wells's like his paper on like forces in like mitosis. So like I've looked at their actual literature, the stuff that they publish, and then I've looked at the stuff that they. So is Dapper. Dapper's done it too. Like,
True.
it really is like I'm sorry, rambling here, but just to kind of finish it. Yes, it is about the data they publish. The reason they don't even attempt to publish these sorts of arguments in actual journals is they know they will not hold up to scrutiny. It's like, it's not just they're confused or they're not understanding why. They know why. I went through 8 blog posts by Gunter Beckley over. Yeah, that's what they are. They're just glorified blog posts by Gunter
They're blog
Beckley
posts with strict formatting requirements.
Yeah, true. Who is an invertebrate paleontologist. I have read his invertebrate paleontology work that has been published, and I have read his work that he writes in Evolution News. If he tried to do in the real journals what he does at Evolution News, that would be like academic. I don't know if fraud is the right word. It's like no one would ever take him seriously again. Like, for instance, when he says like one specific example, he talks about this paper that was published in 2015 on Icaria, which is this late Ediacaran stem bilaterian, and he says, "Oh well, why didn't they consider it a protist?" Now he says that in Evolution News. However, if you go read the paper on Icaria, and he cites a source, he says, "Well, here's a source that agrees with me." The researchers who wrote that 2015 paper cited that exact source and discounted in their paper why the trackways produced by protists are not similar or not similar enough to the trackways produced by Icaria. Like, that's insane. Like, trying to pull that kind of shtick in a journal would be insane. That you get called out so fast. But that's what they do. They do that sort of stuff. Anyway, I'll let you have the last word, and then we'll
one quick interruption from me and a spoiler. The book we're talking about, the commercial for that book and where you can buy it will be shown immediately after this conversation.
That's right. Yes.
True.
Okay. All right, Terry, say the last word, and then we're gonna cut it.
All right. Well, I think that's a complete misrepresentation of the entire situation there.
Okay.
And the reason why is just um you I will read your book, but in your little excerpt there you didn't actually present any reason of why their data itself is wrong.
What are you talking about? I gave you
just
an
said
exact
no.
example.
No, but
Jackson, Terry is giving the last word.
I'm sorry.
Shut
I'm
up.
sorry. I'm sorry. I will shut up.
You did give a singular example of someone that's clearly a fraudulent person that isn't a proper scientist, and using that as an example and applying it to all of scientists that are in a specific field that happens to be the same one he's in, I think that's a misrepresentation of them, because the vast majority of the data is actually sound. And I would say that the main reason why it's not in the mainstream public literature has a lot more to do with the mainstream literature bias than anything else. And it usually is met with with all due respect, with the hand waving and gesturing that you just presented to it, is that it's like, oh well, no, it's just crackpot scientists and it's not good data and vice versa. But you didn't actually provide any reason why that's true. And it kind of just sounds like you're trying to dismiss the evidence. And I mean that in all respect, but it definitely seems a lot more like that to me than, this one specific sect of scientists, because they label themselves in a certain way, are all wrong all the time and they're all fraudulent and et cetera, et cetera. So that's all I would say about that. Just to kind of clarify my position on that.
Okay. Thank you everybody in the live chat who was watching. All now 36 of you. Why haven't you liked this stream? What is wrong with all of you? Thank you, Peter, for hosting. I appreciate it as always. Thank you, Terry, for coming on today and being here and being on the show. I appreciate it.
Thank you for having me.
Of course. And thank you, Dapper, also for being here. I appreciate it.
Thank you. Thank you. Hey, before you leave, I just want to take a second to thank my patrons and channel members, especially those pledging $20 or above. Benthoven, Florevara, Tapioca Weasel, Whispers, Eleron Teller, Horseflesh, Ian Chen, Kelvin Brostek Van Manen, Landon Knoll, Mabdy Babdy, Sphincter of Doom, Wallace, John Ackerman, Ita V, and Strawberry Vein. It's because of my channel members and patrons whom you're seeing on screen that this channel can stay afloat. Without you, it would all shut down. If you want to join the team, there's a link to join the channel below this video, and there's a link to join the Patreon in the description. On the Patreon, you can get a 10% discount for pledging annually, and either way, you get early access to virtually all of my scripted videos, often 3 to 5 months before they come out for the general public. Thanks for watching.